Dear students, in the last lecture, we discussed about blood grouping, right, and genetic cross between two different blood groups. Now we are going to discuss about sex linkage, but before moving to sex linkage, I have this activity for you. That human earlobe, you can check here that two kind of people you will see in population. When you check in this, you can see earlobe fixed and another one is free, right? So check here and do this. Human having either free or fixed earlobes. Count number of free and fixed earlobes in class in which character is dominant. So do this now, check your own and send me a chat so that I check how many we have free earlobe and how many we have fixed. After that, I will reach to a point that if, for example, number of free earlobes more, so it means in this population we have dominant allele, that one, then we will select for that dominant allele. Like for example, if you want to give to that E for free, like if free earlobe is capital, so we will then keep it like E for capital E for that dominant, right? So I hope, I know that free earlobe will be more, so that we will select what dominant, while another one we will select recessive, so I can take it small e. Okay, so let's see students, and I just want to focus those having fixed lobe, because we know already that usually in population free earlobes are more, while fixed earlobes people are few. So let me check that. Give me reply in chat. Most of you having free, good. Who want with any student free, mostly free, right? Earlobe off, okay. Anyone having fixed, give me reply in the chat. Most of you free. Good. It means why am I having fixed? Good. Any other have, please? Give me reply. Most of you having free, right? Who having fixed? Check. So it means we have, up to now, we have only one fixed, right? So for that reason, I will select that one, that allele. For that, I will select recessive, because you know that is less, the number of that fixed earlobe is less in population. So you can take allele like this. For example, if the total strength of the class you have 24, so for example free we have 23 and only one fixed, right? So choose a letter to represent the dominant allele. So dominant I am representing, it's my choice by which I am selecting, but usually we are using capital letter. So earlobes, right? So E for capital E means free earlobe I will select, right? Is it clear? And small I will select small e. I hope this is clear for the recessive one. Now, genotype, the type of allele that organism has, and phenotype, the observed character that influenced by gene. Now you can see here, this E actually, this is our genotype, right? While, you know, the earlobe that's fixed or free, that is phenotype, okay? Sex linkage, right? What is sex linkage? Okay, we know up to here we study about number of chromosome in human cell. So total number of chromosome in human cell 46, right? Now out of these 46, 44 number of chromosomes. 44 number of chromosomes means 22 pairs. Okay. Now the remaining 2, it means 1 pair. These 22 number of chromosomes are actually autosomal chromosomes. Autosomal chromosome. Autosomal chromosomes means these chromosomes concerned with our body, general body characters, control our body characters. Right? While this one, this remaining one, these 2 chromosomes are 6th chromosome. These show 6 of an organism. These chromosomes control 6 related character, right? But along with 6 related character, these chromosomes also control some body character, general body character. So it is not only like that, 100% these chromosomes are concerned with only 6 characters, 6 related characters. No. Some body characters, some genes are also there in these chromosomes that can control some body related character. But these 2 chromosomes, most of the gene on these 2 chromosomes is related with your sex. Okay? Now, these 2 chromosomes in female these 2 chromosomes are similar. Okay? These 2 chromosomes are similar, the last pair, right? I like to just write same is that. Consider if these both are same chromosome. Okay? Structure approximately, you can make it more clear to be more approximately same, right? So these 2 chromosomes are similar in female. That's why these 2 chromosome name given to it XX chromosome. I hope up to here it's clear. While in female, while in male, these 2 chromosomes, one is same just like that is in female, right? While another one chromosome is small, not similar. So to these 2 chromosome name given what? So this one X and this one Y. Okay, so it means XY. So it means these 6 chromosomes in male, the condition is XY. While in female, okay, while in female, these 2 are what? These 2 are XX. In female, these 2 we have X and X. Right? Now, those genes, what is actually 6 linkage? Those genes that are present on six chromosome. That gene is called what? Six-linked gene. That are six-linked gene. The phenomena is called six linkage. Now, what are six chromosomes? We discussed already. H means from here I know that those gene, if gene is present on X, R, and Y chromosome, so that genes are called what? Six-linked gene. Right? Now, let's suppose if a gene is present on X chromosome, so that gene is actually six-linked gene, but which six-linked gene? X-linked gene, because it is present on X chromosome. Now, let's suppose if a gene is present on Y chromosome, so that is called Y-linked gene. That is six-linked, but which six-linked? Y-linked gene. Okay, now if this gene is controlling a specific character, it means that character is related with six. It's six-related character. Okay, now as I discussed with you already that this gene control, six related character, but they can also control some general body character. Let's do example of that. Okay, so examples of six-linked disease are hemophilia and color blind. What does it mean? First, hemophilia, what is happening in hemophilia? The clotting factor or the protein that is responsible for blood clot. Remember, clotting factor. One of the clotting factor that is responsible for blood clotting, right? And that protein gene mutation will come, and that is unable to do blood clotting. So from a very minor injury, blood will not stop. So that condition is hemophilia. So the gene for this clotting factor is present on X chromosome, right? So the gene for that is present on X chromosome. That is responsible to code for clotting factor protein so that they do blood clotting. But because of mutation, due to some reason, this gene, normal gene is what, for example, I am considering normal for H. Normal, it is capital, right? But due to mutation is changed in the normal sequence of DNA. Mutation, what will happen? This gene change will come in this gene, and this gene will convert into what? Mutated form. That will not code for this clotting factor. This will not code for this normal clotting protein factor that is causing blood clotting. So this capital H will convert into small h. This will become a recessive. Is it clear? Now, so when it becomes recessive, so this H will not code for this normal clotting factor protein. Okay. And if a person having this allele, he will be unable to do blood clotting. But here we have still again two conditions. It means if this is working, so blood clotting will happen. Remember, blood clotting will happen. Your body will clot the blood. If it is capital, if that is, in mutated form, if it is in the form of this H, small h, so no blood clotting. Blood clotting means stop the blood. After NG, suddenly after some time your blood will stop. Clot will form, right? So that will not happen. So now if it is X-linked, the dominant convertant recessive. So this is X-linked recessive disease. This disease is X-linked because the gene is present on X chromosome, and this is X-linked recessive disease. First, definitely this is 6-linked, but in 6-linked, which one? X-linked. Okay, now let's suppose if a person having this condition, if a female X chromosome, right? So in her having this possibility, H and small h means dominant and recessive. This condition is what? Heterozygous. Two different alleles are present on chromosome. So this is dominant over this H. That's why the expression will be normal. And this person is not hemophilic. The person is normal. He is not hemophilic. That female is not hemophilic. But this female is carrier. What mean by this is a new word for you? She having no symptom of that disease. She having no symptom of that disease, but she having the ability to transfer this defective gene to her offspring. She having the ability. So carrier is the person who having no symptom of that disease, but she can or he can transfer the defective gene to another person. So he is carrier. For instance, we have another female, these two, and she having this condition, capital H and capital H. So she what? Completely normal. While we having this condition for male it should be XY only. XY, let's suppose a male having H capital, so that male is normal. Am I clear? Now, let's suppose if male having, because these two are not homologous, so one side only H needs, no on another side for the expression. X, Y, that gene or allele is that is recessive. So this person is what? This male having hemophilia. So in this case, no carrier for male. That may be if male having defective gene, so the male is what? Having hemophilia, diseased person. If no defective gene, that male is down. Is it clear up to here? Now we can do the cross for these is the same way that we did for blood grouping. Okay, we will do that. For example, if carrier of hemophilic, right, female married with a normal male, so what should be the offspring probability? So on the same way, we will consider that genotype on the same. How? Let's see. My question is, Carrier hemophilic female, right? One side I'm taking for female, for example, H and small h, right? She married with a man, he is normal. So what should be the probability of offspring? So for this, you have to take first gametes. Am I clear? X and okay. And for this male, you can take two types of gamete. Make a table. Okay, one side male, another side female. One side for male, this side for female. Put the gametes XH and Y and XH capital and X, right? So for example, this gametes fuse with X, right? This one with this. So XH and XH. Now this one with this. So XH and H small, right? Now this Y fuse with this gamete, so XH Y, and this Y fuse with X small h. Is it clear up to here? Now check the offspring. So this one is what? This one is normal hemophilia. This one, she carrier. She normal male. Sorry, he normal male. And this one, disease are hemophilic male. Is it clear? So you can check here, normal, female, right? Carrier female and normal male and hemophilic or disease male. So it means there is a type approximately 1, 1, 1, we will get different offspring. If female having female carrier of hemophilia and she married with a man with a normal male. So that should be the probability of her offspring. Is it clear up to here? I hope this is clear, right? Any question? All right, boys. So our topic of discussion is what? Color blindness. As we discussed already about hemophilia, that is X-linked disease. So color blindness is also what? Inherited disease, and this is X-linked. And X-linked, this is also X-linked disease. What mean by this? It means, you know, first of all, we should know about color blindness. It's a condition in which a person having problem to differentiate between colors. He can't differentiate between specific colors, like especially red, blue, green. He having problem to differentiate between these colors. Okay, there are different types of color blinds on the basis of this differentiation, but we will not move toward that side. Just for time being, just keep it in your mind that for the differentiation of this color, we having special cells and your eye, right? And in that cell there are specific gene, and that gene, you know, if mutation happen in that gene, so a person will be having a problem in differentiation of colors, right? And that condition is called color blindness. Now let's suppose the normal gene for that is dominant. If normally this dominant gene is expressing, so a person will be normal and he having no problem in color differentiation. No, he will be normal, right? While if due to some reason this dominant gene convert into recessive gene, because of mutation, this dominant gene convert into recessive gene, so this person will be colorblind. Okay, now as we know that each organism, like there are within cell, there are gene and pair. So let's suppose if a person having this condition, so this is one allele, this is another. Both alleles are dominant. If both alleles are dominant, so this person is what? This person is normal. He is not colorblind. Both allele will express, it means like in this condition, this capital or this dominant allele will express, and they will make normal protein, and because of that we can differentiate. While for, let's suppose if a person having, right, this one is female, right, because XX chromosome for female. This again female, but in this female, this gene is, this allele is dominant and this is recessive. The condition is heterozygous. So this female normal, but she's what? Carrier. She's normal, but she's carrier. It means there will be, she having no problem of that. She having no problem of color differentiation. But she having this defective gene, she can transfer to her offsprings. It means carrier is a person having a defective gene, but no symptom of that defective gene. And she can transfer that defective allele to her offspring, or he can transfer. Is it clear? So this again for female, X small c and small c, consider it small c and small c both are small. So this condition is homozygous recessive and she's color blind. Okay, now for male, you know, male having 2 chromosomes, X and Y chromosome for 6, right? So we know that color blind is X-linked disease, so that allele or that gene will present on X chromosome. So here I will put it here. What? For example, this is capital C, so it means this allele is dominant. If this allele is dominant, it means this person is normal. Am I clear? Now, let's suppose if male having X and small c means that defective gene, that recessive gene, that is means the dominant gene converts into recessive one by mutation. So it means this person is what? This person is color blind. This male is color blind. Up to here, this is clear, right? If you want to take some hints, you can take. All right, students. In the last lecture, we discussed about what about genetic 6-link disease. So today we are going to discuss about pedigree charts. So what's that? Pedigree is a chart of the genetic history of family over several generations. For example, if you want to show the genetic history of for example a family having color blind disease or having other genetic related disease, and you want to mention that in a form of chart in a easy way, and so that you can understand that easily, and you can make a link between, like for example, if the father and mother having that disease, or one of them having disease, so you mention the others, you mention then their offspring through symbols, and then if that offspring, from that offspring they marry with someone other, so what should be the probability of that disease in other offspring? If you mention that in a chart, in a flow chart diagram, through using special genetic symbol, so that is called what pedigree chart. So we having special sign for that, so let me discuss that. First thing is, for female, we will mention round, and for male we will mention, right? For female we will mention round, and for male we will mention in the form of square. Now if you check it here, if this round is female, so if it is not it's not colored. Colored means like if it is, I mean that if it is blank, so that female is not affected with that disease. That female is not affected with that disease, right? All right. Now, if she's affected with that disease, so that should not blank. You should color it round with color, right? And if he is not infected, so then it should be not colored. So this is for male, and this round is for female. Okay, so it means this female, you can see here, this female is infected, this female, this one here affected female, while she's not affected because it's not colored. If I draw that round and I colored it, I fill that inside, so it means she's affected with that disease. For male, we will mention with this sign. So it means male is not affected. But if male having that disease, so we will cover it complete. Is it okay? Now, let's see about how we will mention these in pedigree chart, right? Now, let's suppose if married couple, for example, this is male, okay, this is female, this is male, both married, okay, so what should be the offspring sibling? So you will mention that in this way. Let me tell you, for example, male, one side I will mention for example, female normal marry with normal male, right? I will mention it like this. It will give me offspring. Then offspring, I have to mention it like this, that for example, all offspring are normal. So I will mention it. If for example, they are twins, so fraternal twin means they are twins but not identical, so then I will mention in this way. But if they are identical twins, so then I will mention it like this. But you will not see many questions from here. It's not, you will not see question from this, but mostly you will mention it by this simple way. Okay, now I hope up to here this is. For instance, we have, if we have carrier, so if female is carrier, so how we will mention that? We will, that round symbol, but half shaded, not full shaded. She's carrier. For male, that sign, but half shaded. Okay. So please mention for carrier, if she's female, round with half shaded. If male, round with, sorry, square with half shaded. Do it like this, boys. All right, boys, let's discuss example. Let's discuss pedigree with example, right? We will take example of hemophilia, right? X-linked disease, right? So H we will select for what? Normal. Capital H for normal and small h for recessive, that is abnormal, are affected with hemophilia. Affected person, right? Now let's do a cross between two people, or between two. One we will select male. Okay, so male is normal, while female have disease. Draw a table for this cross. One side male, another side female. Gamete for this male we know already. This and this gametes, right? For this female, H, one, this male, this side, female. Now, I will put it here, XH gamete for male and Y, and X small h and X small h. So when I do a cross, so XHX, this one with this one with that. Now do for the Y, X small hy and X small hy. Okay, it means two females are what? An offspring that are, the condition is heterozygous, but they are what? Carrier of hemophilia. Two we have carrier. Okay. Now for male, two male we have, and both have what disease? Two male we have, and both are affected with hemophilia. So how you will represent this in pedigree? So let's do that. First of all, you will mention the sign for this male. So male is normal in this case. So for the male, we know we will mention it square, but without shaded. Okay, this male marry with female. She affected with hemophilia. So female with round but shaded. Mention their offspring, please. An offspring to female are what? Carrier. So both are female. First of all, I will mention with round. They are carrier, so I will mention, I will half shaded. While for this female also half shaded, because we have two female and both are what? Carrier. But if you mention the one, also no matter in pedigree. Okay, for example, if you just mention carrier female half shaded, so it's one also enough, no problem. For male, we have two disease, right? Sorry, we should mention it in the form of square, and both are what? Both having disease. This is pedigree. Now let's suppose if this male or this carrier female marry with a normal male, or if this carrier female marry with a normal male, right? A normal male. So definitely up to here this finish, right? Now if you want to do another cross, so let's suppose this carrier female marry with another normal male. So what should be their offspring? So do the cross between them again. For normal male, so normal male HY, okay, cross XH and X small h, because here female carrier. So what should be the offspring? So XH, XH, right? You can also draw table and this X with that. This Y with this XH, and this Y with this XH small. So it means this female is what? Normal female. This is what? Carrier female, and this male is normal male, and this is affected male. So mention that in pedigree, the offspring of these two will be, this is normal female, so mention with normal, round, no shaded. Here carrier, so mention it how? Round female, because X and X female, so mention it round with half shaded, because she's carrier, and carrier we are mentioning like this. And XHY, this male is normal, so just mention it square without shading. And this one, this is what? This is male, but male is what? Having disease. So I have to mention that square complete shaded. So and so on, you can do many cross by using these signs and cross. Okay, from this pedigree, you know that what should be like probability of the offspring. I hope this is clear to you all. If you're having any question, you can ask. Dear students, in the last lecture, we discussed about sex-linked disease, right? Those genes that are present on X or Y chromosome, if mutation happen to that gene, so you know, like disease can be happen. The example was color blindness, and another one that we finished about hemophilia, right? For that two gene, for that two disease, the gene present on X chromosome. Now I discussed with you already that along with 6 chromosomes, for example, this is our cell. So here we have 46 chromosomes. Out of these 46, 44 chromosomes are what? Autosomal chromosomes. They are controlling our body character. right? The remaining two chromosomes that represent our sex, that represent sex-related character, that is X and Y, right? In male, if it is in female, X and X. Those genes that are present in X and Y chromosome, that are called X-linked gene, or if some disorder, some disease happened because of that mutation in that gene, so that is called sex-linked gene. Now we have another condition that is autosomal recessive disease. Means the first one means like the problem will come on autosomal chromosome, the remaining 46. Any chromosome, if some problem happen, right? So we will call that autosomal disease. Now, why I use the word here recessive? Recessive means for that the gene, that gene normally is in dominant form, like in form of capital, like dominant form, but you know, that gene are usually dominant in dominant form, but because of mutation, when that gene convert into recessive form, so that recessive form, because of mutation, if that allele convert into recessive form, we call that autosomal recessive disease, because the actual allele was in dominant form, but now it convert into recessive form, and because of that, we having a disorder, we having a disease. That's why I will use here autosomal recessive disease. Now let's move toward that. I will select two examples for that. The first one we have sickle cell anemia. And sickle cell anemia, actually mutation will happen on chromosome 11. Chromosome 11 having specific gene, and that gene responsible for coding of protein that's making hemoglobin. We know hemoglobin, for example, it is a red blood cell, right? This is a red blood cell. So red blood cell contain what? Hemoglobin. And the hemoglobin is actually the combination of globin protein plus iron, right? So that gene code for this protein, the protein that is present in hemoglobin. So if mutation happen to this gene that is present in chromosome 11, right? So as a result of that, this red blood cell will become sickle cell. One example is sickle cell anemia. Another example we have cystic fibrosis. And cystic fibrosis, you know, this is also hereditary disease. And cystic fibrosis, actually, you know, this is, there is a, in this condition, liquid mucus will accumulate more in your lungs, right? And that can block alveoli or bronchiole, and there will be problem of breathing, and even infection can happen. That mucus element for a long time in that. And then cystic fibrosis also, you know, in that a person can also feel a problem in digestive system, especially because of the mucus, the duct that is coming, like, you know, that is coming from pancreas to small intestine. So that duct can also be blocked, right? Now, and cystic fibrosis actually happening, this problem happening with chromosome 7, right? Chromosome 7, actually, you know, chromosome 7 present, in chromosome 7, there is a gene, and that gene actually code for a special protein, right? And that protein is present in the membrane of a cell. That is called transmembrane regulator protein. The function of that transmembrane regulator is to control the flow of iron from the cell. The but I will not move in detail in that because that is not the topic of discussion. But for time being, just keep it in mind that that protein controls the flow of iron from the cell. Okay? Now, if this flow of iron disturbed, so outside mucus will accumulate more. And that condition is called cystic fibrosis. So the gene that is responsible for that transmembrane regulator that is present on chromosome 7, if mutation happen to that gene, so that condition will lead to cystic fibrosis. Now let me discuss about sickle cell anemia in detail. Sickle cell anemia. Hereditary disease. Hereditary disease means because of mutation in gene and that can transfer to next generation. As the name indicates, in this disease, red blood cell will become sickle shape. For example, this is red blood cell, so this will become sickle shape, just like crescent shape. The word given sickle is a special, it's just like a special, what we can call it, any instrument, right, that we are cutting for the grass. The shape of it is just like this. That's why the name given to it is sickle shape. Okay, crescent shape. Why this problem will happen, I will discuss it now. While anemia means, in this case, abnormal red blood cell will form. So because of that reason, we have a deficiency of normal red blood cell. And a person will be like, pale color appearance. Now, if this red blood cell is converting into sickle shape, why this happens? Let's suppose this is our red blood cell, right? And this red blood cell contains many hemoglobin. And this hemoglobin consists of what? Globin protein, right? Globin plus iron. Both form hemoglobin. Now, this definitely, if it is protein, so protein contain amino acid. A protein contain amino acid, so behind amino acid, definitely the sequence of that amino acid coded by DNA. Right? So for example, this is that sequence of that amino acid. Right? These are different amino acids. So what happened? The gene that code for this sequence of amino acid, one of the codon actually, one of the codon and one of the, inside one of the codon, one nucleotide change. One nucleotide change. Because of that one nucleotide mutation, what will happen? This normal sequence of amino acid will change. Is it clear? This normal sequence of amino acid will change, and instead of glutamic acid amino acid, instead of glutamic acid amino acid that is normally present in this chain at a specific position, let's suppose this is that amino, that will change into valine. Right? And because of that, this hemoglobin, this red blood cell structure will change into sickle shape. Now, when it becomes sickle shape, the gene, the gene actually, its code for it, the normal gene was, for example, I will represent that normal gene by for example, what I can select it here. The normal was, for example, A, right? Or you can represent it with capital S also, no problem. So this A, this normal gene that was dominant in normal condition, that become what? Recessive. That allele become what? Recessive, and that convert into A. Is it okay up to here? Now, up to here, I hope this is clear. Right? Let me explain once again. As I'm telling you that this is the normal sequence of that globin protein, right? For example, this is that amino acid sequence. So instead of one amino acid, the actual amino acid replaced by another amino acid. The actual was glutamic acid, but in that change, another amino acid comes. For example, this is the normal sequence. So this is, for example, one amino acid here. So instead of this amino acid, another amino acid enters. Because of only single nucleotide mutation. Only one nucleotide changed because of that one amino acid completely changed. Right? And because of that, one allele that was dominant that automatically converted to recessive. Now, what the problem these people will face? Remember, the first thing is this red blood cell becomes sickle shape. When it becomes sickle shape, normally red blood cells are flexible. They can, they can squeeze and they can flow to the capillaries easily. But when it becomes sickle shape, so that lost that flexibility. So it's hard for them to be passed in capillaries. Second thing, the shape becomes just like sickle shape. So because of that, they can stick on each other and they can block capillaries. So even this sickle shape also lost the oxygen that it can't transport enough oxygen. So it means they also lost the ability to transport of oxygen and also lost, because of that a person will feel having breathing problem, a person having sickle cell. Right? They can feel pain and there are different pale in color. These are the symptoms of sickle cell anemia. Now, let's just discuss this topic genetic point of view. We have, as I discussed with you, that we have two alleles now. One allele, I will represent it by A, or you can also take it as, but A is easy for me, that's why. So A, let's suppose I am selecting normal, right? And small a, I am selecting what? Affected. Now, definitely within individual, gene is impaired, right? Specific gene is impaired. So let's suppose this person having this condition. So it means this person having two dominant allele. So this person is what? This person is normal. The condition is homozygous. It means in his body, normal red blood cell will form. Now let's suppose I have another condition in which this person having this condition, now it is heterozygous. This is which condition? Heterozygous. So this person is, in this case, he's a carrier, but here is just like, this is something other, he's a carrier, but in this carrier, you can see here, let me write it here. Carrier is a carrier, but in this person, two type of red blood cell will form. Also, what? Also this will form and also this will form. Also round shape and also sickle shape. So it means in heterozygous condition, these both allele expressed. And we know that if in heterozygous condition, both allele expressed, that condition is the example of what? Codominance. So it means that in this person, both type of red blood cell will present. That's why he's a carrier, because he can't feel the symptom. But he will feel only at the time when he climb on a mountain and there is deficiency of oxygen. Usually in life, he will not feel the symptom. That's why he's a carrier. He having a defective gene, he can transfer to other. Right? But still a red blood cell, normal red blood cell is less than that of what it's supposed to be. But because of the presence of normal cell, because of the presence of normal allele and that is expressing, so because of that he will not feel that symptom well, but when he go to a site when there is a deficiency of oxygen, then he will feel that. Right? Now, let's suppose another person I have and he have both gene, both recessive, both defective gene. So in that case, what will happen? In that case, that person will be what? He is affected. Right? Am I clear? So I can represent his red blood cell. All are crescent shaped, and he is the affected person. He will face problem. Right? Now, usually these genotype we are expressing like this. Hey, HbA, Hb per hemoglobin, HbA. HbA and HB. Whenever this codominance word is coming in any place with any cross, so usually we are writing that in a superscript form. It's like A on up, right? Now, and this one we will express it like HA. Am I clear up to here? Now, all the thing is ready. We can do a cross between individuals. Let's suppose if carrier of sickle cell anemia marry with another partner having with normal one, okay, with a normal person. So what should be the probability of offspring? So I will take HbA, definitely this is carrier. So in case of carrier, genotype should be what? HbA and HB, right? Heterozygous condition. Marry with a normal person. So HBA and HBA. Now I will make a table. One side mention male, one side female. So from this, let's suppose if a person get this allele, definitely from this cell we're having two types of gametes can form, right? Here, like for example, we can select it if a person receive this allele, definitely it will be in the gamete, it will be transferred to from our OR because the chromosomes are present here. So HBA and HBA, right? And here HBA and HBA. Do a cross. How you will do a cross? This HBA first with this and then with that. So put it the value here. HBA and HBA for this normal, right? And this HBA and HBA. It means these two offspring are normal. Am I clear? All right. Now do a cross between these two. So it should be HBA, right? HBA and HBA and HBA. HBA small, HBA capital. Right? Now let me check again my cross. HBA, 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 correct? HBA, 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 here should be HBA, HBA. So this should be small. This one with that. And HBA with HBA. So it means these two are what? Both having, the condition is heterozygous. So these two are carrier of sickle cell anemia. These two offspring, carrier. While these two offspring are having what? These are normal. So the ratio with me is normal, carrier, and disease. For normal 2, carrier 2, and disease 0. I hope this is clear. So normal to carrier ratio we having 50-50. Boys, look at here. This question I'm checking Amar Abdul Wahab question. Okay, you can see here. In this case, as I discussed with you already, this is DNA, right? RNA. Now, normally the RNA sequence should be like GAG. That is the codon that code for glutamic acid, and that glutamic acid is present in this normal protein. Right? Now, because of single nucleotide, a mutation, because of single nucleotide mutation, what happened? This normal T, you can see here, this normal T and this DNA sequence. This normal T, this one normal T, like CTC, this convert into CAC, right or no? When it convert into CAC, so definitely a messenger RNA will form from that. When messenger RNA from that, so they become GUG. So it means only one nucleotide mutation, A convert into what? Sorry, T convert into A, and because of that, the RNA sequence also changed with one nucleotide, and that RNA that were coding for normal glutamic acid, that's now code for valine amino acid. So instead of glutamic acid, valine entered to a change. And because of that, the normal hemoglobin structure convert into what? That. Now, your question is that cell definitely, if a person having, for example, HbA, this one is normal, right? Normally, it is like this. But another allele originate from this A because of mutation, and that becomes small a. Now, if capital A is present and this small a is present, both will express because that will be codominance. So it means a new allele come because of that mutation. I hope up to here this is clear. So the topic of discussion for today is cystic fibrosis. As I discussed with you already, it is also autosomal recessive trait or autosomal recessive disease, autosomal recessive. Or we can also say non-6-linked recessive disease. Non-6-linked mean like the gene that is responsible for this trait that is not present on 6 chromosome, X and Y that is not present on this. Non-6 recessive. Okay, now what will happen to, actually the gene that, first of all, you should know about, for example, this is our cell, right? In this cell, in lungs, right? This cell having channels for ion that control outflow of ions, right? So normal the gene that code this channel is actually transmembrane regulator protein. This is protein actually, right? Transmembrane regulator. The gene code for this protein is dominant normally and present on chromosome number 7. Chromosome 7. Normal gene update is what? Dominant. Is it okay? Now, normal gene update is not kept dominant, present on chromosome 7. Because let's suppose I will mention this by F, normal gene. Because of mutation, this will change into recessive form. Because of mutation, when this change into recessive form, right? So this transmembrane protein will be affected, and this will not control, the flow of ion will disturb. Okay, and when that flow of ion disturb, so what can happen? Because of that, mucus accumulation will take place. Mucus accumulation will take place. Normally, the ion should go out from the cell, right? So normally it should be happen like this. And as a result of that, when, when outside ions more, so they can take what can come out from the cell or outside, right? So the outside, the condition will be like not, mucus accumulation will not take place there. But I'm not going to go in detail to that. Just here, I want to tell you that this transmembrane regulator will disturb. And when it disturbs, that can't control well that movement of ion. when it can't control the movement of ion, so as a result of that, mucus accumulation will take place outside. For example, this is that mucus. And when mucus accumulation take place outside, you know, this usually happen in lungs, in the duct of pancreas that is secreting enzyme to our pancreatic juice toward the small intestine. So that duct can block, kidney, bronchioles, etc. can block, right? So this person then having problem in breathing, and even, you know, if it can disturb your digestive system, right? Even this can also disturb reproductive system, right? Reproductive duct, they can also block, okay, if this cystic fibrosis happens. Now, let me summarize once again. Channel that is responsible for the control of ion in the cell called transmembrane regulator. The gene for that is dominant normally, present on chromosome number 7. Because of mutation, that gene will convert into recessive form, and that can't control well the movement of ion from the cell. When it can't control the movement of ion from the cell, as a result of that, mucus accumulation will take place outside. And that mucus can block your lungs, that can block your pancreatic duct, and that can also reproductive duct, that can block it. Okay, now for this we have two conditions. First is I discussed with you, one is dominant, another is recessive. This is the normal form, dominant. While this one is recessive, mutated form through which disease can happen, this disorder can happen. Now, for example, a person having this condition F in F, so that person is what's normal. The condition is, in this case, it is homozygous, dominant, and that person is dominant, that person is normal. Homozygous, dominant, and that person is normal. While if a person having this genotype, so this case, this is heterozygous condition. This person is also normal. Because of this expression of this gene, because of the dominancy of F, the recessive will not express. While if a person having F in F, so the condition is homozygous, recessive. So in this case, this person is affected. It means this person having cystic fibrosis. Am I clear up to here? Now let's suppose we are selecting two parents, one having carrier, both carriers, right? That are normal but carrier, that's both having defective gene. So if we are going to do a cross, now if you do a cross, so what should be happen? First of all, you can check it here. This one, you can see here, for this we have two gametes, F and F, and for parent 2, this gamete and this. So means like two gametes from parent 1 and 2 from another. Now let's do a cross. If this, if you can also draw a table for this, okay? If this, if you do a, if this, if fuse with this, if, so what will happen? Normal person will produce. So this person is now normal, right? So this person is now normal, this person. This one person is normal right now. So up to here, if we cross this F with that, so this person will be normal, okay? Then this F with with what? With this app, right? With the carrier one, with this app. So this person is what? Carrier. Is it okay? Now, first of all, what I did, this F, capital F with capital F. I got two capital F and capital F. Both are normal. Both are capital. It means this person is normal. Then this F, this capital F cross with this small f. So you got what? F add small f. This person is carrier, right? While come toward this one, please. This one cross with this one first. So you got it what? F, capital F and small, carrier again. And this one, this recessive cross with this recessive. I got this one. You got it? So it means this person having cystic fibrosis. So we got two carrier, one normal and one having cystic fibrosis. Carrier is a person having defective gene and he can transfer that to new generation. Am I clear up to here? Boys, let me discuss to you this once again. Transmembrane regulator, that's protein, the outflow of ions, right? This ions from the cell, it is taking out. Why? You know, if extracellular space outside, in extracellular space ions are more, so it means outside the solution is concentrated, and from this water can come from cells or from a near area to that place, and that, you know, in that place no mucus will accumulate. Right? Now, if no ion is getting out, so what will happen? Outside, mucus accumulation will start. If mucus accumulation starts, so in that place, bacteria growth can also happen. So because of that, you know, infection will happen in that place. So more chances of also infection in cystic fibrosis. Protein synthesis, actually, you know, conversion of DNA code into sequence of amino acid. That is protein synthesis. You know, like in protein there are amino acid. Let's suppose this is amino acid 1, this is 2, this is 3. So each protein having specific order of these amino acids. This order code by whom? This code by DNA. And I discussed with you already that for each amino acid there are three nucleotide form one codon, right? That code convert into this amino acid sequence. All amino acids arranged on specific sequence. For example, here is amino acid 1, here is amino acid 2, right? For example, this is valine, this is glutamic acid, this is tryptophan, like different amino acid we have. So this sequence code by whom? This code by DNA. Now, if you check another protein and that, for example, and that the sequence is just like this glutamic acid, and then another amino acid like methionine. So that sequence is different than amino protein X, for example. So this sequence code by DNA, and conversion of DNA into this sequence of amino acid, this is actually called what? Protein synthesis. Now, we know that, for example, this is a cell, okay, and this is the nucleus of the cell. In the nucleus of the cell, we have what? DNA, right? So this DNA, well, first of all, when it's converting into that sequence of protein, it will convert into messenger RNA. Means the DNA code will be reached by whom? By messenger RNA. And that messenger RNA will then come out to the cytoplasm, and that will bind with ribosome and will convert into that sequence of amino acid. Let's suppose I'm taking this DNA out. So for example, this is that DNA, right? Now this DNA, will uncoil. This will open that place which have to be read. So that open, right? And from one strain, messenger RNA formation will start. From one strain, what will start? Messenger RNA formation, like this. For example, here we have in DNA, here we have adenine. So here should be what? In RNA, there is no thymine, so here should be uracil. If here is guanine, so here should be cytosine, right? If here is, for example, here is what? Thymine, so here should be adenine. So like this sequence of this messenger RNA will form. Now this messenger RNA will now come out in A12, for example, this is that messenger RNA. So this messenger RNA will, this is that messenger RNA. This will now move toward the new cytoplasm by passing through the pore of nuclear membrane having pores. Okay, small holes, through that it will come out to the cytoplasm. Right now, this messenger RNA is right now here. Sorry. So this messenger RNA is right now here outside in the cytoplasm. Right. So this messenger RNA have to bind with the ribosome. Ribosome is a cell organelle that help in the protein synthesis. When it binds with ribosome, so my ribosome will read this sequence of messenger RNA. So let's suppose this sequence is just like this, UCA and so on, right? So definitely this UCA code for a specific amino acid. Let's suppose this UCA code for amino acid X, and here is another codon, this amino acid, that code for amino acid Y, and there is another codon, that codon means triplet nucleotide sequence, that code for amino acid Z. So this sequence will convert into what? To amino acid sequence. Am I clear? And this definitively, if it become long chain, so that is now what? It is protein. Is it clear? So the first step is actually conversion of DNA into what? Into messenger RNA. This DNA convert into messenger RNA. Okay, when it convert into messenger RNA, okay, that messenger RNA passes through nuclear pore of the nuclear membrane of nucleus and reach to cytoplasm, and within cytoplasm it's bind with ribosome, and ribosome convert that, like read that sequence of messenger RNA, and when ribosome pass through that, am I clear? So ribosome read that sequence, and on the basis of that sequence, different amino acids come together and form what? A protein. It means this code provide what instruction for which amino acid should be come first, and then what should be another, and then which should be another. So a peptide linkage will form between all amino acids, and that will convert into, that will become a protein, right? So in this case, you see a code for G. Let's suppose here is another codon CCC, so that code for X amino acid, and here is another codon UAG, for example, so that code for Z amino acid. So for each amino acid there are different codon, but we are not going to the name of that amino acid. Okay, here I just take it as a supposition. Suppose XYZ are amino acid. Okay, now this messenger RNA will come to the new cytoplasm, and it will bind with the ribosome and will convert into what? Protein. It's not like this messenger RNA will convert protein, but ribosome will come and will read that sequence of messenger RNA, and on the basis of that, different amino acid will combine together and will form protein. Okay, now this is your protein. Is it okay? This is protein. Now, you know, this conversion of this DNA into messenger RNA is called, conversion of DNA into messenger RNA is called what? Transcription. Is called what? Transcription. Transcription. It means protein synthesis. The first step is transcription, in which DNA convert into messenger RNA, while when messenger RNA is converting into protein, that is called what? Translation. It means protein synthesis will complete in two steps. The first one is transcription and another one is translation. And transcription, DNA is converted into messenger RNA, while in translation, with the help of ribosome, messenger RNA will convert into protein. Is it clear? Okay, boys. So in this case, the DNA act is a template for the formation of messenger RNA. And when messenger RNA form from DNA, that will move to the cytoplasm through nuclear pore. And that will combine with ribosome.